No, my haramo, welcome. I'm very excited today to introduce today's speaker, who is Dr. John Ramana. Um, I am really excited and looking forward to John's kōrero today. John is an early career researcher, obviously with Bioprotection Aotearoa. He's currently co-leader of Understanding Drivers of Plant Health with Professor Ian Dickey, and he is also going to talk to us today about his mahi on microbial indicators of forest health and environmental change. His work spans multiple spatial scales. So what that means is micro to macro, I guess. Um, and he is working integrating field studies, microbial DNA sequencing, and satellite-based remote sensing to uncover the ecological patterns that shape forests across diverse environmental conditions. John is a local to Ōtautahi, fabulous place, Christchurch, having completed his postgraduate studies at University of Lincoln and PhD at University of Canterbury. But I'd actually like Dr. John, to give us his own introduction. So kia ora, John. Really looking forward to it. And welcome. The floor is yours.
Kia ora, kia ora. Kanui te koa me te hari ki te ia koutou. It's so nice. It's a joy and pleasure to see you all, albeit online today. Thank you for making the time to come out. Kia ora, Nick, for that introduction. I think I wrote half of it. No, I'm kidding. Yeah, like Nick said, I'm a local to Christchurch. I've managed to do research here. And so after my PhD at Canterbury, I did a postdoc with Bioprotection Aotearoa at Manaki Whenua Land Care Research with Dr. Kate Owen and Ian Dickey, and that's going to be some of the work I'll be talking about today. It was from the last couple of years. So yeah, let's dive into it. Like I said, this work was done at Landcare with Dr. Kate Orwin and Professor Ian Dickey here at UC. And so kind of around this idea of microbes being a really dynamic kind of indicator of potentially forest health and also environmental change. They can tell us a lot about what's going on in the environment. So when this project started initially, it was kind of looking at this idea of what is ecosystem health, and that it's quite a like a buzzword, gets thrown around a bit. It can mean a lot of different things, and it's really hard to kind of it's easy to say ecosystem health, but really hard to define what we actually mean. Because as a scientist, you're kind of looking at it, it's a really holistic term. It's integrative of different things, really ambiguous. But one thing that's, I guess, really important when we talk about ecosystem health, forest health, and things like that, is to acknowledge that disease and death are a part of a healthy ecosystem, right? These are completely normal processes that occur. And a couple of years ago, we were out with a bioprotection Aotearoa hui in the Banks Peninsula and on a boat. And this is on near Akaroa on our way. And just from the boat, you can kind of see these patches of dying trees just kind of scattered around. And it kind of got us talking and thinking, and the question was raised, how many dead trees in a forest until it's a problem, right? Because death, again, is a normal part of forest ecosystem processes, but when does that become an issue? When do we need to start taking notice? And the problem is, well, not a problem, but a tendency is that we ignore pathogens, right? Until they can't be, until a pandemic breaks out, until it's quite often too late, really. When you so you know we're not actively studying things until they're a problem, and so a natural tendency is to study disease and sick and dying individuals and ecosystems. So quite often when say like myrtle rust breaks out, we'll go and we'll check out what's going on in those diseased ecosystems. But studying the sick and dying can only tell us how to not be healthy, right? It doesn't actually tell us what are the things of a healthy ecosystem, a person that prevent them from being sick. And so I guess healthy ecosystems aren't ecosystems without disease, but they're without pathogens, but rather they're healthy ecosystems are ecosystems without the presence of disease. Um, And so it's really important for us to study this baseline, right, of health. Just so that we know what's healthy, and then when things go wrong, what to look for, how have things deviated from the normal. And so by studying and focusing on healthy ecosystems, we can begin to ask some interesting questions. Things like, is there a core microbiome of a healthy ecosystem, right? What does that look like? Do all healthy forests kind of share a core component or microbiome that allows them to remain healthy? We can ask questions like, what are the level of pathogens that's normal in a healthy ecosystem, right? Again, developing that baseline, because when things go wrong, when pathogens get out of control, we kind of know what was there to begin with and how things have shifted, rather than being kind of on the back foot and retrospectively trying to figure out what's going on. And along those lines, I guess, also, how do the communities of beneficial and pathogenic microbes change with climate? Like, how resilient are our forests to climate change? What will happen in 50 to 100 years' time as the climate changes? Will we see how will pathogens change in the course of that time? Will trees still be able to associate with beneficial microbes that allow them to thrive, or will those associations be lost or altered? And so there are a lot of interesting questions that we can first ask to develop this baseline, to better prepare ourselves for when something does go wrong, or when climate extremes do happen, right? And so I guess that's kind of the conceptual framework of the project. And we decided to do this in Kānuka as a model ecosystem, Kānuka forests on the Banks Peninsula. This is obviously a Kānuka tree. It ranges from like these large bushes to really monodominant tree stands across the peninsula. We chose Kānuka, sorry, Kānuka. I hope I said Kānuka. We chose Kānuka because it's a really important tree species for native restoration and reforestation. So it's an early succession, like a pioneer species that comes in first, grows, and then allows some of those longer living native trees to come up through under it. And then those forests are going to last hundreds and hundreds of years. And so it's really important for native restoration, really important for carbon sequestration. And biologically speaking, it's actually quite intriguing because and I'll touch on this in the next few slides, is that Kānuka is actually a dual mycorrhizal plant species. So uh, I'll dive into that, what I mean by that, a bit further in the next slide. But so the work that I'll be talking about today is kind of three different studies and kind of looking at three broad groups of microorganisms. The first being fungi. So obviously fungi, are, not obviously, but I think they're the most important group of microorganisms. Um, but they range from really beneficial, like saprotrophic fungi that break down nutrients, to mycorrhizal fungi, really beneficial type of fungi, to plant pathogens that can be quite detrimental. Swinging back around to mycorrhizal fungi, so there are two types of mycorrhizal fungi, generally like two main types really. Ectomycorrhizae. These fungi form these fruiting bodies and quite often mushrooms that you see, and they are associated with plant roots where they form a symbiotic relationship and give plants nutrients and acquire carbon in return. So those are ectos. And there are also arbuscular mycorrhizal fungi. So these fungi don't create these fruiting bodies that you kind of see. They don't make mushrooms, but they infect root cells. So this is a root that's been stained. And they infect root cells and make these little tree or bush-like structures. And that's where they exchange nutrients with the plant host in return for carbon that the plant's fixed. And so that Latin word arbusculum means little tree or bush, and so that's where arbuscular comes from. So with these two main types of mycorrhizal fungi, most plant species will either associate with one or the other, but very rarely do plants associate with both, or very few species do. And Kānuka is really interesting because it's one of those species that simultaneously associates with ectomycorrhizal fungi and arbuscular mycorrhizal fungi at the same time. And that's really interesting ecologically as well, because there's opportunities here for trade-offs between different strategies. So these fungi offer slightly different benefits. And so Kānuka, as part of its method of being resilient to climate change and potentially pathogens, may have this ability to trade off between the two to kind of survive. So in addition to fungi, we also looked at bacteria. Again, bacteria range from highly beneficial, like nitrogen-fixing rhizobium, to decomposers, and they can also be really pathogenic. A lot of pathogenic bacteria around, and again, they're really widespread globally. They're everywhere, found in really extreme and harsh environments, and are just widespread globally. And the final group of microbes that I'll be talking about today are oomycetes. Some you might have heard of them referred to as water molds. These are much less well known about. We don't know too much about them, but what we do know is they can be major drivers of plant disease. So Kauri dieback is a Phytophthora, which is an oomycete, and so it's a really big threat to our native forests right now, right? So they have a huge impact on forest and ecosystem health. They're also actually really common in fish and aquatic life. Just as a side note, like if you ever had a goldfish and it's kind of flipped over and white fuzzy stuff comes out, that's an oomycete. I found out really recently, and that definitely has happened to me. Um, so yeah, those are kind of the three main organisms that we'll be touching on today, and they kind of fit into these three different studies, but are all linked to the big study. And so I guess the three questions we can ask that I'll be asking from these studies is the first one being, how will oomycetes, and these are the potential plant pathogens, how will they change in diversity and composition with climate change? Because it's really important, like I said, for us to establish a baseline. There's not too many studies about oomycetes in New Zealand. There are a few, but we don't have this really comprehensive baseline of what's already there, so that when pathogens like Kauri dieback start coming in, we're aware of kind of what's there to begin with to see what's changed. Maybe it was already present in the environment and just a change in climate's kind of triggered this outbreak, right? Like, so we can answer a lot of questions if we know what's there. And in the same way, we can ask, how will climate impact the beneficial fungi associated with Kānuka? So those mycorrhizal fungi, those dual associations. Is Kānuka adaptable to switching between the two based on what kind of climate it's in? A lot of those basic questions. And then finally, we can kind of take a step back and zoom out and kind of look at the microbiome as a whole and ask this question of, are there microbial indicators that could predict tree health? Like, is there something healthy trees have in common, or diseased trees kind of have in common, right? And so those are the big three studies that I'll quickly be just sharing some brief results with you today. Um, so to do this, like I said, we sampled Kānuka stands. So the sampling we did was stratified based on elevation and rainfall. And all that means is that first, over here, we identified all of the Kānuka stands on the Banks Peninsula. So this is, wherever there's Kānuka, this is where it is mainly. And then we mapped out for each of those stands its elevation and its mean annual rainfall, and kind of plotted those. So each of these black dots is one of these stands. Then we split them up into bins, and then randomly tried to sample from as many bins as possible, where there were Kānuka and where we could get obviously permission and things like that. And so those red dots are the sites that we selected. And so we selected 30 sites all across Banks Peninsula. And the reason for the stratification of sampling is that we really wanted to tease apart the role of rainfall and elevation, and to get a really wide range of environmental predictors, because we're going to use that as a proxy for climate change, right? What happens when temperature goes up in terms of elevation, and you get a lack of rainfall and things like that. And so you can see here that we have sites that are pretty high rainfall, but really low elevation. And then equally, you get the other end of the spectrum where high rainfall, but also high elevation. And so it really allows us to tease apart the environmental drivers that are associated, that influence the microbiome of these Kānuka trees, and kind of just hone in on what's left. And this is how they kind of fit out on the map, roughly. And so you can see that spatially, it's a pretty good spread across the whole Banks Peninsula. So at each of those 30 sites, we sampled Kānuka stands at both the community and focal tree scales. So we sampled two trees at each site, so one on the edge of the forest and one in the interior. You can see here, this is our focal tree, and we've set up a 10-meter plot around it. So we sampled soil from various areas around the plot. We sampled the leaves of other plant species in the plot, so including these little shrubs you see down here on the ground to other leaves from other Kānuka that happen or other plant species that were in the plot. And we also sampled the roots of the main Kānuka tree as well as the leaves of the main Kānuka tree. And what that allowed me to do was to get an idea of what's on the roots, what's on the leaves of the main tree, and kind of what's around it, what's it being influenced by, what's the potential pool of microbes that it can pull from or share or kind of spill over into or back and forth. And so, yeah, we sampled this and sequenced the DNA from the soil, leaves, and roots. Um, and so, firstly, let's look at oomycetes and to see what we find. Just to get an idea of what kind of oomycetes uh, are in these substrates of Kānuka, because we don't know a lot about them. So again, really important to establish this baseline. There's heaps going on in this graph, so just bear with me. Um, all it is that each of these bars is a particular oomycete, and the color represents the genus that it belongs to. And the length of the bar is how abundant it was in the particular substrate, so soil, roots, or leaves, or pool leaves, right? Like I said, heaps going on, but there's just a couple of things I want to draw our attention to. The first being just interesting that there's this really dominant Aplonopsis. We don't know a terrible amount about this genus, but it's interesting that it's so dominant in soil roots and the community of leaves. The other interesting thing is that we see a really high abundance of Phytophthora in Kānuka roots, right? So Phytophthora, like Kauri dieback is a Phytophthora. So these are plant pathogens that we're seeing a really like high increase in some of the roots. And also really interesting is that the leaves, foliar samples, so the leaves of Kānuka and the leaves of other plant species, have a really distinct community of oomycetes that's not present at all in the roots or the soil. So it's a really specialized community, kind of dominated by Perennispora and Pythiums, a bit more around here. And finally, this is probably the coolest and most interesting uh, point is that the most dominant oomycete on Kānuka leaves and leaves of other plant species in our native forest is completely unidentifiable. Like we have no idea what it is. The DNA sequencing as part of the study, we did a lot of phylogenetic analysis. Ian really led that and some really intense stuff. And even with doing all of this phylogenetic work, we couldn't really place it into any of the big clades. So yeah, we have no idea what it is. Um, but it does show that there are really, in terms of developing baseline, that what substrate you look at really does have a really distinct community of oomycetes and potentially pathogens. If we look at the richness, if we look at the number of species we find, there are some really interesting trends in the community samples. So just looking at these two graphs on the left-hand side here, on the y-axis, we have the species richness in pool leaves and soil. And on the x-axis, we have elevation. And you can see this like downward trend that the number of oomycete species drops off as you increase in elevation. So higher elevation sites have fewer oomycetes, right? And so I guess that's a good indicator of climate and how they're responding. But if you were to put it in terms of a climate change perspective, an increase of 3 degrees Celsius is the equivalent to about losing 428 meters in elevation. So, say with warming, you might be going from sites that potentially at the moment only have about maybe 2 or 5 species of oomycetes to now being in about the 20 to 30 range, which is really significant and a big deal in terms of the potential health of our native forests, because we're going to start seeing potentially start seeing an increase in potentially pathogenic microbes that are going to start coming up into the soil. So the big kind of takeaways for that were that the strong effect of elevation suggests that oomycetes will likely increase in frequency and richness with rising temperatures. So that's something to be aware of. And also that the most abundant oomycete on Kānuka remains completely unidentifiable. That's interesting, but also a really good challenge and a really good next step for us is to try and figure out A, what is it? And B, what does it do? Is it harmful? Is it not? What conditions does it become harmful in or not, you know? So it's a really interesting kind of result to build on for our next for the next few years of work. And if you want to read a bit more about the oomycete part, that's just been published recently in Fungal Ecology. It's free to read, and I'm sure the links are up on the bioprotection page for this project and stuff like that as well. So yeah, it's free if you want to go and read a bit more about what we did. Um, cool. So the flip side of oomycetes are the beneficial fungi, the mycorrhizal fungi that I talked about. And so if we look at how they change in terms of climate, it's really quite interesting. So first, let's look at those ectomycorrhizal fungi. So these are the ones that make the fruiting bodies. So we see that the composition of ectomycorrhizal fungi in the soil is correlated really heavily with elevation. So on the y-axis is a measure of the fungal community. It's a bit arbitrary, but on the x-axis elevation, you see this really tight correlation. So um, and then when you look at arbuscular mycorrhizal fungi, so these are the ones that penetrate the root cells, you see that it's not soil, but rather root fungal communities that are correlated with elevation again. It's important to note here that there was no correlation with elevation or rainfall with species richness. So the number of species of mycorrhizal fungi is relatively the same in regards to elevation and rainfall at least. But what's interesting is that the composition of those fungal communities is shifting, right? So what's the abundances of what's there is quite dynamic and responding to the environment and the soil for ectos, but actually in the roots for Kānuka roots for arbuscular mycorrhizal fungi. So the number of fungi are the same, but the composition of those fungi and the roots are different, suggesting that Kānukas may be able to kind of select fungal partners based on the environment that it finds itself in and respond to those dynamic changes, and could be an indication of its resilience to climate, I suppose. Um, and so, yeah, those are the kind of two big takeaways from this was that the beneficial fungi in soil and roots are responding to environmental change, to those gradients in elevation and rainfall. The composition is shifting, but not the number of species. So unlike the oomycetes, we're not losing species or seeing an increase in the number of beneficial fungi in the soil, but rather they're shifting in terms of their composition. And that we're seeing that Kānuka is potentially selecting arbuscular mycorrhizal fungi based on its environment to help it be more resilient in whatever environment it tends to find itself in. Um, so those are oomycetes and mycorrhizal fungi. Like I said, we don't know a lot about either of those two in terms of Kānuka, so really important to first establish that baseline of how they change with the environment. But then now we're going to take like zoom out a little bit and look at the microbiome as a whole to see if we can identify kind of general patterns that might be linked to tree growth. Um, and so we're using tree growth rate as a proxy for tree health. And to do that, you kind of need an indication of how old the tree is that you're sampling and also its growth rate. And so to do that, in addition to taking all of those soil and leaf samples for DNA sequencing, we also cord these trees, which is a non-lethal way of aging. So you drive this thing into the trunk and pull it out. And then you get a little tree core like this. You can then mount it onto some wood or something like that, and then sand it back with some really fine sandpaper. And you can begin to see really clearly these rings and start beginning counting the age under a microscope. Once you look at it, you can actually see the individual cells of wood and how they change, and you can really like form some distinct lines and start counting how old these trees are. So yeah, that's how we got an indication of tree age. And then if you measure the DBH or the diameter of the trunk, you get this growth over the lifespan of the tree, right? A tree's growth rate. And so what we did was we just simply paired tree growth rate with the microbial community to see if there were any correlations that stuck in terms of what are really decent predictors of tree growth rate. Um, what we found was quite interesting. We found that the soil and leaf fungal diversity showed contrasting relationships with tree growth. So um, on the y-axis here is deviation from expected growth. All that is that growth rate, the tree growth rate that I was talking about, but accounting for the environment that that tree was in and the fertility of the soil, right? And so seeing if the value is positive, it means that that tree grew more than we expected fertility. But if it's negative, then that tree grew less than what we thought it would based on the environment it's in. And on the x-axis here, we've got soil fungal richness, so the number of fungal species in the soil, and leaf fungal richness. So yeah, for soil, you see a positive trend, and that kind of makes sense. It kind of follows the narrative of soil biodiversity is a really good thing. And so the more fungi you have in the soil, the better the trees are growing. But if you look at the leaves, it was quite interesting that you see this opposite trend that actually biodiversity isn't a good thing. And it could be an indicator of something that's going wrong, right? And so the more fungi you're finding on the leaves, the more poor the tree growth is there. If we dive into this a little further, we see that those patterns are largely driven by saprotrophic fungi. And that makes total sense for the soil, right? Because these are the fungi that are involved in breaking down uh, organic matter and releasing nutrients. And so obviously the more fungi you have doing that, different species, the more you could expect tree growth to benefit, which is 100%. But then for leaves, again, it's that same opposite pattern. And it could just be that trees that are growing really poorly have damaged leaves that are exuding some kind of substance for saprotrophic fungi to kind of latch onto and feed and get opportunistic, right? And so that could be one reason why we're seeing this uh, opposite trend with soil and leaves. And then generally speaking, bacteria followed a really similar pattern where leaf bacterial richness was negatively associated with tree growth rate. So again, it kind of points to this idea of potentially damaged leaves attracting opportunistic bacteria and fungi, and you see you see the number of species elevated and in richness because of that. So the main kind of takeaways from this were that Kānuka growth is strongly linked with soil and leaf microbial diversity. But it's really important where that biodiversity is, and it's crucial to understand if those relationships are positive or negative with growth, right? It's kind of it goes against this notion of biodiversity is always good and always needs to be protected, but actually, yeah, it is good in the right context, but actually could be an indicator of something not quite right, depending on where it is. So yeah, those are kind of the big the three big kind of studies that we kind of came out of this work. Yeah, establishing baselines for pathogens and microbes, and also just finding indicators of tree health with the wider microbiome. There are a lot of people to thank for this work. Obviously, Ngāi Tahu, and particularly the relevant representatives from Wairewa and Kokorara Tarunanga. The many landowners that allowed us to sample on their land, including Christchurch City Council and DOC, and a whole bunch of people that helped with field work, sample processing, acquiring funding, and yeah, just a ton of people to thank for this work. Um, and yeah, thank you so much for your time, for making the time to come and listen to me. And I'd love to take questions and definitely get in touch with me. I'd love to chat more about any of this stuff. Yeah, yeah.
Thank you, John. That was fantastic cordial. I'm very excited to see that you're really exploring deep diving into the microbiome of our taonga, Kānuka.
Mm.
And I was really, really interested to see how widespread Kānuka is on the on Banks Peninsula. So is
Yeah.
that a is that is I guess it's all recovering or regenerating, isn't it, from a
Yeah,
widespread clearance. So what you're looking is that reestablishment of a native ecosystem. Um,
absolutely.
so I was quite interested in that dominance. Is there a particular type of Kānuka related to that eco type or the that rohe or that area of Banks Peninsula? Because I know there's various ecotypes around the country, but
Mm-mm.
is there a specific one or is it just the general one that we know and love, the common taonga?
I think it's the common one. Kunzea ericoides
Right, yeah.
is the one that I believe. I don't know. I know there's some contentious renaming. But I mean that's what we're kind of using it as at the moment, yeah.
Yeah, kia ora. And also just reminding everyone, links to John's recent paper, congratulations, and his research site that co-leading with Ian and his whole research mahi and articles are there in the chat for your further reading. So now some pātai. Um, Kia ora e hoa. Great kōrero, Hanarea. Great kōrero, John. Did the high number and abundance of uomycetes in the pooled leaves relate to the height of the surrounding plants, i.e. being closer to the ground? And that sort of...
Hmm. I haven't looked into correlations between height and the number of room seats, to be honest, but the way we sampled was to kind of try and avoid that bias of different heights and different plants. So we first made a list of all of the plant species in terms of their abundance in the plot, and then just went down the list, took a sample from each of those species so that we weren't overcompensating based on abundance. So if something was really abundant, it wasn't that we were sampling, say, out of 100 whole bunches of leaves, 99 were like grasses, right? You know what I mean? So we did try to kind of stratify that sampling based on species like diversity as well. So I'm hoping that would have hopefully buffered out some of those differences. But that's a really interesting point, and we definitely have the data to check that.
Kia ora, Hanarea. And supplementary question. Um, also, did you analyze the microbiome of those Kānuka cores, actually? Because they look really interesting in themselves as well.
Yeah, yeah, they do. We haven't done that. I don't know if that's something one of our PhD students we've just hired will look into because their role is to kind of look into the pathogens of the past, and they're going to be using pollen cores to kind of see what fungal pathogens have been there. But and potentially we talked about the idea of maybe using the tree cores too. Yeah.
Yeah, there's always more mahi to do, isn't there? But it's a great biological resource there.
Mm.
Kia ora, Alice. Did you consider sampling planted Kānuka sites? Oh, that's a good point. Do you have any speculation about what you could find in regenerated tree land? So I guess that whole re natural regeneration versus, us
Yeah.
giving it a go and an
Mm.
anthropomorphic help re-veg
Yeah.
project.
Yeah, yeah, yeah. So some of the sites were planted. They were landowners who had undertaken a little small restoration effort. Most of them weren't. At the moment, actually, we have a PhD student starting as part of this next tranche of funding who's asking that exact same question of looking at natural versus planted restorations, and actually incorporating nurseries into it as well to see what pathogens are coming from the nurseries into restoration plantings, and then B, comparing the prevalence of those pathogens to native like natural regeneration to see kind of how long are these pathogens going to stay about? Are they kind of spilling over into natural systems? So that's quite an interesting bit of work that's just starting up, but definitely looking at that aspect of planted versus natural regenerated bush. Yeah.
Kia ora. And also, no mai kia ora, Kare, Becky Ganley. Great talk, John. Fascinating research. Tautoko that. I know you're not going down to the individuals, but I was wondering if you'd found Phytophthora Canovia. It's an interesting species and fits with your work. A top unwanted pathogen overseas, but here in Aotearoa, it infects radiator, radiata pine, and but in our natives, it's only been found peripherally. For example, in Cody a couple of times, it seems to have the impact on Pinus radiata and not other species. So, was that one of the species you found?
Off the top of my head, I'm not sure. It won't be hard for me to look it up, and I will. That actually does sound really interesting. I will take a look in the next couple of days and get back to you, Becky, because yeah, that's quite interesting. Yeah, it'll be easy enough to find out.
And if yeah, and that's a very good point because it's yeah, there's that whole harboring pathogens and inoculum sources be quite interesting. So kia ora, Becky. Kia ora, Edward. He is asking in the last graph, it looked perhaps like tree growth followed more of a curve where there was less growth and at the extreme low end of leaf fungal activity. Then say that tree growth peaked at a moderate amount of fungi before dropping off as fungal activity reached a maximum. Could some of the leaf fungal activity be needed and beneficial to the actual trees themselves at archive? So good, that's a good point.
Yeah, really good point. I'm just trying to, sorry, I'm just going to reread that. Followed more of a curve where there was growth, at the extreme low end of leaf fungi, then tree growth peaked at a moderate. Huh. Yeah, yeah, potentially some non-linear trends there, which I'd have to look at. But also interesting. Yeah, you're right. Could some leaf fungal activity be needed and beneficial to the trees? Yeah, perhaps. But I would, when we did the Umai study, we found that our like success
Yes.
of detecting Umai was far lower in leaves, like something stupid, like 50 or 60% of samples failed PCR, and we did a whole bunch of stuff to verify if it was just the lack of Umai or if something went wrong. But it was indeed the lack of Umai. Um, uh, and so I think partly that antimicrobial component of Kānuka leaves might play a really important part. And I found that quite interesting that even with the antimicrobial kind of the essential oils and what have you, that you still see such a high diversity of fungi and bacteria in those leaves at poor growth.
Mm. Yeah,
Mm.
kia ora. Um, Kia ora, Suman. Again, accolade to your wonderful talk. It's quite interesting to see Kānuka roots being a potential reservoir of Phytophthora, given that it's a native plant and historically coexists with Kauri. Do you think that could be a potential correlation?
Yeah, yeah, potentially. I mean, the jury's still out on whether Coccidioidex a native pathogen or not, right? Like there's good
Mm.
evidence both ways. And yes, certainly maybe uh, Kānuka is a reservoir of and potential pathogens. But again, it's that's part of that developing a baseline to know what's there to begin with, so that when something does go out of whack, you can kind of see, okay, the environment's pushed this into the push this pathogen into becoming more of a problem.
And just to total Becky around the nativeness or otherwise of the
Mm,
Phytophthora canovia, it's really interesting that whole native versus exotic, but
yeah.
definitely Canovii seems to be on that native end of it. And I guess some of those species, those unknown species that you've found,
Mm,
I recall some work by the late Ross Beaver and Simon Coleman, his master's student, that were baiting waterborne phytophthora or oomycetes actually from the catchments, and were finding that good old pond scum or water scum, and finding the just what was Simon always said the justice for the ecological oomycetes that are just there, benign or
mm,
in a place and space in the ecology of water and obviously terrestrial environments. And so, of course, doesn't mean they can't go out of whack or out of into a negative deleterious impact, but that
mm,
whole framing of what's there, as you say, the baseline is so critical. So
yeah.
I think your work on those Kānuka leaves and others is going to be critical in helping answer those sorts of questions.
Mm.
And kia ora, Simon. Have you tried any isolations of the leaf surfaces themselves, acknowledging some might have culturability issues?
Yeah, absolutely. We haven't, and that part of again this next thing of funding is to actually try and bait some from the leaves and kind of see. But yeah, like you say, it is quite hard. We had a summer scholar try this, I think actually last summer, and it was quite unsuccessful.
Hmm.
So we're trying to think about ways around it, or yeah, even if it's sequencing really like tiny fragments of leaves, like very minuscule, to potentially hopefully only get one or two really good long reads out of it, or things like that. But yeah, it's yeah, the isolations weren't terribly successful, I think.
Hmm. Yeah, and also culturability is a factor because there's all those intricate types of leaf baiting and types of media that can
Mm.
coalesce or bring out those as pure isolates has often been the difference. But at least with what you're doing, you know they're there to start with. So and you get some clues to from their identities as then how to isolate them. Jenny just also endorses Becky's interest around that
Mm.
Phytophthora canovio question, which is a clearly a
Mm.
key one for many people, including industry and others. So yeah, kia ora, Jenny, definitely. Obviously that's a real hot issue of your mahi. Um, One thing I keep those pārtai coming, everyone. If you have any supplementary questions or new questions, please put them in the chat. Or if you don't have time, you obviously can contact John directly now because you've got all of the links to him. What do you feel now as a key area you and Ian in your mahi around this are
Hmm.
going towards? Like what are some of the key clues of what of indicators of forest health that you're finding that you'd really like to pursue now? Because obviously this is hugely wide
Mm.
topic or kaupapa that you could go in 101 directions, but
Yeah.
where do you see now as what you'd like to investigate next as part of that whole answering some of that?
Yeah, so definitely, like I mentioned, we're really keen on finding out about what pathogens are coming out into our natural ecosystems through nurseries. That's a big avenue at the moment, and also looking at this idea of what we've coined as the pathogen tax. So, like the idea that yeah, pathogens are a normal part of ecosystems, but actually, how much are they impacting productivity? If you were to ask a farmer, like for example, like a pathogen's a big deal, and he'd probably say no, and then but you'd probably ask him what he's doing, and he's spraying a few times a year, he's taking time out, and even then you're losing maybe 10% of crops, but you kind of live with that level of loss in a sense of it's normal, but actually, could you quantify that of what is the effect oomycetes and pathogens are having on our native ecosystems? So looking at seedling germination and growth, and forest decline as well. And so that pathogen tax idea is something that we're going to be looking at in a lot more detail and trying to develop a bit more. And also, nowadays, the use of remote sensing to identify pathogens early. So quite often, it's hard to get boots on the ground in some of these places, especially in areas like the peninsula where there's a lot of lot of like hard bush and terrain to get through. So are there ways that we can kind of ID and what I'll be working on is IDing hotspots or patches quite early on that are going through decline to then we can go in and ground truth some of that work to see if actually yeah is that an issue is what we're seeing from these satellite images yeah reliable could be used future to ID outbreaks really early.
Yeah. No, I mean that will be very fascinating because
Mm,
as I said, it's a really key area of and taking us into new, highly innovative areas that we don't know much about our whole taonga and our environment. So like all the best for that. So
mm,
kia ora, thank you. We wish you well in that. Um, Without any further comments, all questions, I'd like to just again, Homai Te Pākipaku, thank you again for that kōrero and reminding everyone that John now joins alongside Sarah, Hanarea, Alexa, and the Rangahau Rising webinar series, which you can go back and see the kōrero by Sarah and Rethinking Biocontrol in the genomics era, or Hanarea's amazing kōrero around Maori Tāwaka, the syzygium microbiome in the context of myrtle rust, and Alexa and her studying the drivers of soil carbon resilience across agro ecosystems, and now John. They are looking at the microbial indicators of forest health and environmental change. So you're joining the legacy of fantastic kōrero, and I really do. If you haven't seen any of those previous presentations in the series, you can see the links there in the chat, or go to Bioprotection Aotearoa's website and click on those recorded presentations. I really do think they're a fantastic resource and shows you really what's going on from our most brightest future science leaders and our co-leaders in tranche 2 of Bioprotection Aotearoa. So kia ora, John. And on that note, moving right along, shuffling papers. I also want to promote next month's Rangahau Rising presentation from Laurelyn Rosagnaud, who works with Philip Hume, and on November the 19th. So next month, same back channel, same time, must register, 1 p.m, so November the 19th. And what Laurel and her mahi is going to talk about is the non-native plant invasion in Aotearoa, insights from the Mānuka and Kānuka shrublands. So it's really awesome how all these presentations are interlinked, and so you'll get to hear more of her mahi and what's happening in those Kānuka shrublands. So just wanted to not only thank John and previous speakers, but also welcome Laurelyn to join the Rangahau Rising next month. So please register, and the registrations are there thanks to Anna and the team. Kia ora. And also just wanted to wish everyone a happy spring. Spring has sprung. And thank you for your time and joining John today. We're really pleased that you came to hear John's whakaaro and his talk around his mahi. So kia ora to everyone who's taken that time. It's really awesome. Spread the word that these are available to anyone that registers. Please, they're a fantastic resource to see the amazing research that's going on in the bioprotection Aotearoa space. So kia ora. And I will close our hui today with karakia whakamutunga. And we'll get going, reminding you that you can see these karakia on the Tukutuku app. So again, download that from your app store. And thank you, John, again. Kia ora.
Sure.
Ka whakaira te tapu, ka wātia ai te ara, kia turuki whakataha ai, kia turuki whakataha ai, ti hei Māori ora. So safe travels, everyone, and see you next month. Ka kite anō. Kia ora.